Hi, my name is Alexa Mogus, and in this presentation, we are going to be discussing the structure of skeletal muscle. There are three types of muscle. There's skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscles make up the muscular system, and they attach to the skeleton. Skeletal muscle contraction moves bones to which they are attached, allowing the body to perform motor activities. Skeletal muscles that support homeostasis include those in acquiring, chewing, swallowing, and those essential for breathing. Smooth muscle is located in the walls of hollow organs and tubes. Contraction regulates movement of blood through vessels, food through the digestive tract, air through respiratory airways, and urine to the exterior. Cardiac muscle is only found in the walls on the heart. Its contraction pumps life-sustaining blood throughout the body. Muscle cells develop tension and shorten or contract by moving specialized intracellular components. Muscle comprises of the largest group of tissues in the body, accounting for half of body weight. Skeletal muscle makes up about 40% of body weight in men and 32% of body weight in women. Smooth and cardiac muscles make up another 10% of the total weight. Controlled contraction of muscles allows for a purposeful movement of the entire body or parts of body. For example, this may include walking or waving your hand. Controlled contraction of muscles allows for manipulation of external objects. So this may be things like driving a car or moving a piece of furniture. The controlled contraction of muscles allows propulsion of contents through hollow internal organs. This includes circulation of blood or movement of a meal through the digestive tract. And controlled contraction of muscles allows for emptying contents of certain organs to the external environment. So this may include things like urination or giving birth. So first off, let's talk about skeletal muscle. So muscles are categorized as striated or unstriated, depending on whether alternating dark and light bands or striations, stripes, can be visualized under a light microscope. Muscles are categorized as voluntary or involuntary, depending on whether they are innervated by the autonomic nervous system and not subject to voluntary control. In this case, skeletal muscle is striated and it is a voluntary muscle. Skeletal muscle is categorized as voluntary because it can consciously be controlled. However, skeletal muscle activity is also subject to subconscious involuntary regulation. This is related to posture, balance, and rhythmic movements such as walking. The location of skeletal muscle, they are attached to the bones of the skeleton. Skeletal muscle is made up of bundles of long, thick, cylindrical, striated, contractile, multinucleate cells that extend the length of the muscle, and they function in movement of the body in relation to the external environment. Cardiac muscle can be classified as a striated muscle and an involuntary muscle. Cardiac muscle is described by an interlinked network of short, slender, cylindrical, striated, branched, contractile cells connected cell to cell by intercalated discs. Cardiac muscle is located in the wall of the heart and it functions in pumping blood out of the heart. Smooth muscle is classified as unstriated muscle and involuntary muscle. Smooth muscle can be described as a loose network of short, slender, spindle-shaped, unstriated, contractile cells that are arranged in sheets. The location of smooth muscles is in walls of hollow organs and tubes, so this can be like the stomach and blood vessels. And smooth muscle functions in movement of contents within hollow organs. So now we are going to talk about skeletal muscle fibers. A muscle fiber is a single skeletal muscle. It is a large, elongated, and cylinder shaped. It measures from 10 to 100 micrometers, And skeletal muscle consists of muscle fibers lying parallel to one another and bundled together by connective tissues. Fibers extend the entire length of the muscle, and it can be seen in the pictures displayed on the screen of a muscle fiber. A myoblast is an undifferentiated stem cell that develops into muscle tissue. Myo meaning muscle and blast meaning a primitive cell that forms specialized cells. So in embryonic development, skeletal muscle fibers form by fusion of many smaller cells known as myoblasts. Cells differentiate to form multinucleated muscle fibers for muscle contraction. There are multiple nuclei dispersed beneath the plasma membrane in a single muscle cell, and there's also an abundance of mitochondria. So what is a myofibril? A skeletal muscle fibers contain many myofibrils that are cylindrical intracellular structures that extend the entire length of the muscle fiber. Myofibrils contain the contractile apparatus of the muscle, and they constitute for 80% of the volume of a muscle fiber. Myofibrils consist of a regular arrangement of organized cytoskeletal microfilaments, thick and thin. Thick filaments are primarily composed of the protein myosin, and thin filaments are primarily composed of the protein actin. The levels of organization of a skeletal muscle include, first off, the whole muscle, which is the organ, a muscle fiber, which is a cell, the myofibril, which is a specialized intracellular structure, thick and thin filaments, which are the cytoskeletal elements, and myosin and actin, which are protein molecules. And in this image here, you can see at part B the relationship of a muscle fiber and a myofibril. And the portion of the myofibril is down at the bottom of the image here. And we'll get into more of the parts of the myofibril later on in the presentation. First off, we are going to talk about thick filaments and myosin. So myosin makes up thick filaments. Each thick filament has several hundred myosin molecules packed together in specific arrangements. So myosin molecules, as seen on the left here in the image, they consist of two identical golf club shaped subunits with their tails intertwined and their two globular heads projecting out at one end. The heads form cross bridges between thick and thin filaments. Each cross bridge has two essential sites for the contractile process. This is known as the actin binding site and the myosin ATPase site. Each myosin subunit has two hinge points, one along the tail and the other at the junction of the tail with the head. A thick filament is made up of myosin molecules lying lengthwise parallel to one another with their tails oriented toward the center of the filament and their globular heads protruding outward. And this is seen in the image B here, which is the thick filament, and you can also see where it displays the cross bridges at the heads of the globular heads. In this slide, we are going to talk about thin filaments and actin. So actin is the main structural component of the thin filaments. Thin filaments contain three proteins: actin, tropomyosin, and troponin. The main structural component of a thin filament is two chains of spherical actin molecules that are twisted together. Actin molecules have a binding site for attaching with a myosin cross bridge. The myosin and actin binding at the cross bridge leads to the contraction of a muscle fiber. So as seen in the image on the left here, we can see the thick and the thin filaments, and that's the cytoskeletal components of a myofibril. And then down towards the bottom, you can see the protein components of the thick and thin filaments, the myosin head, myosin tail, and the thick filament. And then you can see the three proteins of the thin filament: actin, troponin, and tropomyosin. And the image on the right here displays a thin filament, and it shows the tropomyosin and the troponin. And we're going to get into those in the next two slides, those proteins. And then you can see the actin molecules, the green, that is the binding site for attachment with myosin cross-bridge. All right, so let's go into a little more depth about tropomyosin and troponin. So contraction does not take place in a relaxed muscle fiber because actin cannot bind with cross bridges. This is due to the proteins tropomyosin and troponin positioned within the thin filament. So tropomyosin molecules are thread-like proteins that lie end to end alongside the groove of the actin spiral. Tropomyosin covers actin sites that bind with the cross bridges, blocking the interaction that leads to muscle contraction. Troponin is a protein complex made up of three polypeptide units. One binds to tropomyosin, one binds to actin, and the third binds with calcium. When troponin is not bound to calcium, the protein stabilizes tropomyosin in its blocking position over actin's cross-bridge binding sites. So essentially, the muscle is relaxed. So as seen at the top of the image here, part A, where it says relaxed, at the first step here, we have no excitation, and then there's no cross-bridge binding because the cross-bridge binding complex site on the actin is physically covered by the troponin and tropomyosin complex. Therefore, the muscle fiber is relaxed. When calcium binds to troponin, the shape of this protein is changed so that tropomyosin slips away from its blocking position. Therefore, actin and myosin can bind and interact at the cross bridges, resulting in muscle contraction. So in this sense, it would be excited. So on the bottom here, we can see the image. So the muscle fiber is excited and calcium is released. The released calcium binds with the troponin, pulling the troponin-tropomyosin complex aside to expose that cross-bridge binding site. Binding of the actin and myosin bridge triggers a power stroke that pulls thin filaments inward during contraction. And this image as a whole explains the role of calcium in turning on cross bridges. Tropomyosin and troponin are also known as regulatory proteins because of their role in covering, so preventing contraction, or exposing, permitting contraction to the binding sites for cross-bridge interaction between actin and myosin. So in this slide, we are going to talk about more of the cytoskeletal components of the myofibril, specifically looking at the A and I bands. So a myofibril displays alternating dark bands, which are the A bands, and light bands, which are the I bands. Bands of all myofibrils line up parallel to one another, producing the striated appearance of a skeletal muscle fiber. And this can be seen in both images displayed on the screen. First off, we're going to talk about the A band. The A band is made up of a stacked set of thick filaments along with portions of the thin filaments that overlap on both ends of the thick filaments. Thick filaments lie only within the A band and extend its entire width. The H zone is a lighter area within the middle of the A band where the thin filaments do not reach. Only central portions of thick filaments are found in this region. And on the diagram here, you can see the little area where the H zone is marked. Now let's talk about the M line. The M line is where supporting proteins that hold the thick filaments together vertically within each stack can be seen. The M line extends vertically down the middle of the A band within the center of the H zone. The I band consists of the remaining portion of the thin filaments that do not project into the A band. And we can see here on the image to the right and left the I and A bands and where they're located. Let's talk about the Z-line. So the Z-line is a flat cytoskeletal disc that connects the thin filaments of two adjoining sarcomeres that is visible in the middle of each I band. And on the image here, both images, in the Z-line area, we can see where the sarcomeres are located. The sarcomere is the functional unit of skeletal muscle, and it is found in the area between the two Z-lines. A functional unit of an organ is the smallest component that can perform all functions of that organ. A sarcomere is the smallest component of a muscle fiber that can contract. So what is titin? So titin is made up of single strands of giant, highly elastic protein. Titin extends in both directions from the M line along the length of the thick filaments to the Z lines at opposite ends of the sarcomere. And this can be seen in the image displayed on the screen. Titin is the largest protein in the body, being made up of nearly 30,000 amino acids. And titin serves three important roles. So it serves as scaffolding, where it helps stabilize the position of thick filaments in relation to thin filaments, contributing to sarcomere stability. Titin acts as an elastic spring. It augments a muscle's elasticity. It helps a muscle stretched by an external force passively recoil to its resting length when the stretching force is removed. Titin constitutes the parallel elastic component of muscle because it behaves like an elastic spring. Titin also participates in the signal transduction pathway. For example, it is involved in the complex pathway where muscle enlargement is in response to weightlifting. Finally, let's discuss cross bridges. Cross bridges extend from each thick filament towards surrounding thin filaments in areas where thick and thin filaments overlap. Cross bridges project from each thick filament in all 6 directions towards the 6 surrounding thin filaments. Each thin filament is surrounded by 3 thick filaments. Single muscle fibers may contain an estimated 16 billion thick filaments and 13 billion thin filaments. And as you can see in this image here, where it is labeled the cross bridges as part of the sarcomere.